morning, everyone. Thank you for the invitation to the conference. This is my second time at NYU. Always a pleasure to be here. Always a pleasure to talk to students. I think it's very important. And I'll give you a little bit of who I am. I'm a Peruvian, actually. I entered the university in 1985, and I'm giving you this date because that was the year PCR was published. And so I was at the university as a student when the paper came out, and the people said, this is going to revolutionize the way we do things. And it's true. And so at that time, I was attending the medical school, not the biology school. So I entered as a student in medicine, and then I decided to leave and then do something different. And that's why I'm in agriculture. And that time, the university, and this is very important for the students, ignite in you certain way of thinking. And because I came from a medical school, the way of thinking was about diagnostics, understanding the causes of diseases, understanding why people got sick. So this was very important to me because when I look at DNA, I didn't look at DNA as just only knowing what's happening with genes, but how we use these genes to advance our knowledge. And so what I'm going to present today is how you use these genes, how you use the information, and why I'm in the way I am. So cassava is a very important crop in Africa. The crop wasn't bred there, wasn't domesticated there. The crop was domesticated in South America, where I'm from. And so the challenge is that cassava takes about 10 to 15 years at best to produce a new variety. And so by the time, and thank you for the introduction about climate change, because climate is changing dramatically fast. And so if we need to adapt food to the climate, to the crisis of the climate, with this rate of breeding, we'll never get there to feed the world. 800 million people feed off cassava. This is the main source of carbohydrates for 800 million people in Africa. And it's the source of income for 1.5 billion people. So it's a very important crop. It's not at the level of maize or soya, because these crops are very important and very well worked. Here, there are no tools, there were no resources to understand how to proceed with speeding up the process of breeding. And so coming myself from the cotton life, where they invested a lot of resources, and that's how I ended in Australia, by the way. I lived 17 years in Australia, and that's where I did my PhD, and I advanced at the beginning of this technology. So when I started, sequencing was done in plates, you know, and you have a software that you run the letters A, C, P, and then you have to start writing and pass many times until you have a quality of the sequence. And today, this happens in a flash, correct? And millions of bases. To the point that even bioinformatics, which was something that I thought I would be getting into, became a science outside biology. Now it's a science of computing. There's so much information, so much processing that you require so many different tools and technologies. So when I joined this team, the question was, how do we get the genome sequence of cassava, which was, by the way, sequenced after the wheat genome, very early, 2009. Many other crops did it very late. So cassava was the third full genome sequence. So then I thought, this is great, because even cotton was not at that level at that time. And the reason why it was sequenced is because the Department of Energy in the United States thought that because cassava has cyanide as a compound, it was a threat to security. So the State Department put the money to fully sequence cassava to understand if someone can breed cassava to become the most toxic poison on the world and kill everyone. So, and that was the driver, but really good for us because we have this fantastic resource. So can we take cassava to the next level? So one of the things that is important to put in context is 20 years took for us to move from 20 tons of the crop a hectare and produce 7 tons of the most important commodity that cassava has, which is a starch, to 30 tons a hectare with 12 tons of starch. So this is 20 years of breeding. And so if we need for cassava to be profitable today, you have to produce not 12 tons a hectare, you have to produce 17 tons a hectare to make a farmer in Africa economically viable. So the gap still, this is 2017, and we are in the run. So how can we make this faster? So we deployed, I developed all these tools that were required in Cassava to move forward. So you needed to do the pre-breeding. So we needed to understand better how the diversity of the crop started, because it's not a crop like maize. You know, breeding is the art of making everything to look the same. And when you have diversity, everything looks different. So you go to a cassava field, and it's so much difference there that you don't know if you're in a forest or really in a cropping field. And the way cassava is homogenized is by clonal propagation. So you get a nice, outlier, and then you clonally propagate the outliers. So you go to a cassava field, and they look beautiful, like it's maize or it's cotton. But it's just made that way because it's clonally propagated. So if you go to a breeding field, it's a forest. Plants that are a half a meter, plants that are 7 meters tall, trees. So this is the way every time you get a cross in cassava looks like. And so the question is why? I mean, why so much variability, and how can we control that variability? So we needed to look for gene identification, the genome size, how to barcode the DNA, how to automate the phenotyping. So all these technologies were required in order to progress in breeding. So breeding doesn't happen today. Modern breeding doesn't happen because I go and select the best plant in the field, basically. And that's what classical breeding is. And everything started with a great resource that is available because of funding given by many good souls, the Gates Foundation, the USAID, the Europeans. So they fund a center, a group of centers called the Consultative Group for International Agricultural Research. And this consultative group, and CIAT is one of the members, preserve hundreds of thousands of accessions from the diversity of many places in the world that are food related. And one of them is cassava. So in this collection, there are 6,640 accessions that come from all over the place where cassava is being domesticated, from the south of Mexico down to the north of Argentina. So this is the span of the area of domestication of cassava. And like you can see in the flowers there, I mean, this is just the cassava tribe. All flowers are different. So you look in the field, I didn't put the plants because it's very difficult to get a good picture that shows the diversity of the plants, but the flowers is a good example of how diverse a cassava crossing block could look like. So what we did is we said we're going to sample randomly at 10% of the collection in order to do a good screening using the sequencing technologies at that time that were available. So we needed to be smart in the use of resources, and then look at how can we untangle the genetic diversity of this. So what we did is having the reference genome. So we use a reduced representation library of the genome doing RADseq, which is a reduced amplification of the DNA, which allows you to do assemblies of very small contigs and then harvest what? Variation, SNPs. And so that required a lot of computational power because we were in the millions of bases. And so to really harvest all that information, we needed to do a very smart way of doing this, and we collaborated with the South Green platform. These are located in France. So it's a very driving group in terms of developing systems for managing data. So we partnered with them, and 71,540 SNPs were untangled out of the cassava diversity. So with that, we did what everybody will do, duplication. How much of the collection is duplicated? What is the kinship relationship so that we can select the most diverse types? And then what the unrelated genotypes are. Because if we want to proceed looking at the discovering genes, we need to work with unrelated species. If one want to do genome-wide association or anything like that, we need to know which were the most unrelated genotypes. So the first thing we discovered is that there are a lot of duplication. So this is an example. What is in blue is the breeding programs in Africa. And there is, at the beginning, every number that you look there is different, but genetically, they're exactly the same, meaning that in the process of selection, they selected many times the same clone. So in the different years, because remember, it's 15 years of work, making mistakes of labeling plants is very, very, very likely, and this is an example of that. Similarly, is the collections that were done in the organization. So you see there plants coming from Costa Rica, Venezuela, Panama, Cuba. So the collectors went and collected without any tools other than just their perception of diversity, collected plants in different places, but at the end of the day, the same plant, the same diversity caught their attention. So we have so many duplicates collected in different parts of the world that allows us now to say, okay, this is the way to move forward because 27% of the materials in the collection are duplicated. So this collection takes $1.5 million a year to maintain. If I can reduce 27% of that collection, I will be saving about $500,000 that can be used for other purposes in science because we need the money that we have to make the resources more available. So this was one of the first low-hanging fruits. 35 of the materials are related in first degree, so meaning that breeding, some kind of breeding was going on already. Some selections in first degree relationship were happening. It means that there is a heterosis going on. So the combinabilities in the first degree are very useful. So that tells us how to move forward in terms of breeding. Sorry, I'm going backwards. Sorry. And 88% of the collection are related in third degree kinship, and only 12% were totally unrelated. So then, with this information, we selected from every group, representative, good representative, and then we ensemble a panel for doing the diversity. But one of the things that was clear from this relationship analysis is that there were two lines, one in South America and one in Africa, that contribute to most of the parentages, and we didn't know that at that time. So Brazilian 20 was the use in the breeding program, but when you look at the records of the breeders, it's not a plant that is being used in the directed breeding. So meaning that the crossing of cassava is also a problem. Means that bees come first in the breeder, and the bees control what plant produces best pollen to transfer across. So because the animals, and this is one of the things that we learned in cassava, the insects were most of the driver for breeding cassava because the insects use the resins, they use the toxins, they use the pollen, they use the sugars to build their communities in the jungle, so where this plant come from. And so that tells you a bit how the forces of nature operate. And then there was a nice also to know that in Africa they used that line that we didn't know where it came from, but was related to Brazil as well. So meaning that was an important germplasm for breeding. And so we look at across the 585 accessions, and we untangled 7 subpopulations in cassava. So what that means for breeding, it means that breeding in one location is not possible, correct? So we need 7 locations, and these locations were actually geographically dominated, as you will see in the next slide. So we know that 5 of those subpopulations were from the north of South America, and 2 were from the south. And then one of them is from Brazil. I mean, the other one is from Paraguay. And then the less diverse ones were from Brazil and Paraguay. Unlikely what you will think that the diversity of cassava was coming from Brazil, as it's being written in many papers. We don't see that much diversity and a very well-structured population. So thinking, okay, what's going on? More diverse and more things were going on in the north of South America. So we could identify quite simply that these populations belong to certain hubs. So you have the Caribbean subpopulation, which is in the driest part of the continent, together with the savannas. The difference between the two is the altitude. So the savannas of Latin America are about 2,000, 2,500 meters tall, whereas the Caribbean are at the sea level. So that was a third difference. But also when we moved to the Andean, in the Andean region, we also saw a subpopulation compared to the Amazon. The difference again was altitude. So the Andean subpopulations were up to 3,000 meters elevation, the ones in the Amazon at sea level. So this altitude was important. And then the humid Atlantic forest and the dry Atlantic forest of the continent, these materials were quite well selected, and we wanted to know, is really Brazil the center of origin? So we ran a model to see where actually cassava could have been likely to be bred based on the geographical position of the lines that were collected. And then the result was that somewhere between Peru and Ecuador, those lines came from. And not surprisingly, most of the genes that we find for diversity in cassava, whether it's insect, whether it's beta-carotenoid content, whether it's peeling capacity, come from those two gene pools. Whitefly resistance, name it. So virus resistant. And so what's the beginning to say? So if we time the time of domestication of cassava, cassava was probably domesticated in the Andean region, but then bred further in the north and south of South America. So moving to the drylands where they wanted to adapt cassava. So then cassava has been adapted to grow in dry conditions. So there was a requirement. And then the history of humanity, if you look, there is a population called the Taino Indians. And the Taino Indians has to move from the Amazon to the Cuba and Puerto Rico. So that's when, Columbus arrived. Columbus arrived and met these Taino Indians. And they were telling the story that they didn't belong to these islands. They have to escape because of climate change. And so they have to readapt the crop to the climate conditions. So cassava has some of the key elements to understand adaptation of crops to the changing climate. And these people did it very, very, very fast. And why? Because cassava creates very good hybrids in first generation. But you have to know what you're doing, correct? And so we selected a panel at CIAT, and you can see clearly this is one location. We selected the 141 most diverse materials and grow it in the location that's supposed to be the best location to grow cassava. And you can see in the panel that some of the lines were not doing that well. And so that tells you that environment plays a big role for cassava. Cassava was bred to be an environment-sensitive crop. And so we then look at flowering genes, high cyanide, because that was important for the quality of food, because it's toxic. And cassava has the advantage that most of the cassava is processed. So, and for the one that is toxic, so you have an enzyme that breaks the toxicity. So it's so clever the way they breed it that even though the crop has high levels of cyanide, it can kill a horse, just one bite of some of these lines. But the minute you break the tissues, it releases an enzyme called linamaerase, and the linamaerase breaks the cyanide and make it volatile, so then it becomes safe to process. Therefore, that explains why in Brazil, for instance, where most of the high cyanide cassava is, they eat pan de queso, they process flour, because that's the way to get rid of the cyanide. And then in the areas of the Andes where it's sweet, the cassava is totally sweet, they boil it. And this is now linking genes to user preference and things like that. So I mean, just to give you an example, so we found, for instance, the cooking quality, high cooking quality of cassava in the Andean material. So you see there very clearly that the Andean material was the one carrying the gene for easy cooking. When we looked at the cyanide content, then where the material is coming from the Amazon and the Atlantic humid forest, which is the Brazilian area, with high levels of cyanide. This cassava is used for processing. When we looked into the easy peeling, those came from the area for Paraguay. And then probably that's the way of making easier for the processing of the crop. And remember, processing cassava is always related to women. So this is a gender-sensitive trait. And so if we know now that one community solved the problem of peeling cassava, if you go to Africa, you can see hundreds of women with knife peeling this very difficult crop because the germplasm they got it from the Andes. So it's not meant for processing. So someone resolved the problem. So we can transfer the easy processing now, and the peeling could be easy to do. And then what we did is we look at an old paper from cyanide to see if can we do GWAS with this panel effectively, so we can untangle the same genes that were published in that study. So when we ran the GWAS, exactly the two genes published in this paper very early in the 1990s came to the two proteins came to be significant in our GWAS study. So we could do it. But genomics goes beyond just genome-wide association. So the other thing we did is we tried to work with biparental populations, so where we know we have contrasting traits. And beta-carotenoid was important because is a crop because cassava is eaten by 800 million people in Australia, in Africa, and these people are minority mostly. So micronutrients needs to be delivered in an effective way. And then one of the ways to deliver micronutrients effectively was to transfer beta-carotenoid, for instance, to cassava. The question is how we do that. And then obviously the most easy trait to follow is, okay, it's orange or not orange, correct? The same as the carrots. And so that's what the breeders start doing. So they start tracking the beta-carotenoid content, and they start making some headways, for sure. So you can see the lines there. So the content of beta-carotenoid, the content of beta-carotenoid was increasing, as you would expect in any breeding line. And so we said, okay, how do we speed the process? We need to discover genes. So 2 groups embark into this process. So we're us at CIAD in 2012 and 2013, where we managed to narrow the area where the QTL, the potential loci involved in this, is located, which is between 23 million base pair and 25 million base pair in chromosome 1. And then we couldn't nominate a gene. And the reason why we didn't nominate a gene at that time was because the phytoenzyme synthase gene, which is logical, was there. But when we did the analysis on the performance of that gene, it was not different between a white root, a orange root. So it was the same level of expression. So it was not, we hesitant to nominate that gene as a gene. So that's what we did. Then IITA in 2014, with a different population, narrowed the gene to the phytoenzyme synthase, but we know it's not the gene. So then, I mean, it's a gene that is required, of course, it's a phytoenzyme synthase. You need phytoenzyme synthase to produce beta-carotenoid, but is this a driver of the increase of beta-carotenoid? It's not the case, and I'll show you why. So if you look at the chromoplast, whether it's a high beta-carotenoid content, you can see clearly the color. You know, the white roots also has the beta-carotenoid. The difference is because it's so much starch in cassava that the white root color reflects the light. So if I look at a white root, the orange is there, but I cannot see it. And this is the difficulty of doing phenotyping. So, and then people said, oh, look, it's white. Yeah, it's white, but there's so much starch that is reflecting the light, I cannot see the yellow. I cannot see the orange. So, and this is the limitations of doing phenotyping by eye. So, and then there was a revealing thing, and so we said, where is those genes? So we embarked into doing transcriptomics. And transcriptomics tells you a bit of the activity of the different proteins. And so at that time, we had to perfect the way we do RNA extraction in cassava. It's a plant with a lot of sugars, a lot of proteins, so very difficult to do a high-quality cassava extraction of RNA for doing true transcriptomics, a big challenge. So we did that. And then one of the things that was clear from that data, if you see here, you can see clearly how the genes were separated. The activity that drives the beta-carotenoid accumulation was something related to stress. Thank you. So then what we know is that there's a regulatory factor that cannot be untangled by GWAS or that cannot be untangled by anything else that is driving this. It's a master regulator that drives the accumulation of beta-carotenoid, but also defend the plant from stress, particularly heat stress, climate change, because these genes are in the core of the genome of cassava. Another gene that was simple for us to do it was the amylopectin-free cassava. And why is this important? Because amylopectin starch is used for thickening. And thickeners are very important in many industries, particularly yogurt. And this is a natural product. So Ingredient funded us to do the research. We found the molecular marker so we can track the mutation very easy. We can also tap into materials that are difficult to tap into. And then now they release these products, which are called green label, clean label, because cassava is grown. We know fertilizers. We know this is a fairer crop. And also, they don't have to use any chemicals to modify the starch to make a good thickener. So this is a win for the cassava community. And so with that, I think I finished because it's the time. I had another story to tell you, but this is the end. So thank you very much for your attention.